Welcome to the first Infogest webinar from the beautiful Emerald Isle of Ireland. My name is André Brodkorb. I'm a researcher here at the Teagasc Food Research Centre in Moore Park near Cork, and I'm joined by Myriam Grundy, our communication specialist who makes sure that everything will run smoothly. And I'm also joined by the two presenters, Myriam Grundy and Uri Lesmes. Okay, so I just have a few slides to start off with, just to tell you a little bit about the Infogest Network. The Infogest Network came out of a EU-funded cost action in 2011. For 4 years it was funded. Subsequently, we went on with some industry funding, and over the last number of years, INRA sponsored some of the costs that came along. We organized a number of international conferences, training schools, staff exchanges, and so on. And since the end of the European cost action, we changed structure, and here is the current structure. The chair of the Infogest Network is Didier Dupont and vice chair Alan Mackey. And we have several working groups. So it's open and inclusive. So you can join at any stage. You can join as many work groups as you wish. We have different themes from different in vitro models, food interaction, meal digestion, absorption models, lipase, starch, and in vitro, in silico methods. These are some of the high impact outputs. It's the simplest model, the static digestion model, was published a number of years ago in Food and Function, and last year in Nature Protocol. Here are a number of other high impact publications, really highly collaborative work, very successful. We published a book on digestion, the impact of food bioactives on health, again, pretty high impact. If you want to join the network, if you want to receive information, you can send an email to Nathalie Lamar and or Didier Dupont, and you find their email address on the website. Just Google Infogest. And recently I create the LinkedIn group. So in LinkedIn, look for Infogest, just follow, and there we keep you up to date. So we have two excellent speakers. The first speaker will be Uri Lesmes. He will give a good overview of the area of plant proteins, and followed by Myriam Grundy. The whole webinar will be 1 hour. The presentations itself have been pre-recorded, and we were on the safe side here, just a little bit conservative, just to make sure that everything works. And the question and answers will be live afterwards. So you can type questions, any question that you have, into the question and answer box, type it there, and we will answer it live. So the first talk is Uri Lesmes. Give me a second.
Hi, welcome to the webinar on food processing, digestion, and trends in plant proteins. My name is Uri Lesmes. I come from the Technion in Israel, and in the next few minutes I would like to walk you through this part of a very hot topic that we see all around us. As we see in supermarkets all around us, there's a growing demand for protein and protein-added products. We can see it in the dairy aisle, in yogurts, in cheeses that are now protein-added or protein-fortified. We can even find it in breakfast cereals and energy bars. We can actually find it in many, many, many products in our supermarkets. In fact, the whey protein market is seeing a 7% annual growth rate in the last few years. So there's a growing and an immense demand for proteins and alternative proteins. In fact, even the scientific community that you are part of has brought all of us to discuss plant-based diets and plant-based proteins. Well, What proteins are on our menu? Well, if you look back a year ago, sometime in May, the Wall Street Journal actually discussed on its front page the actual launch of Beyond Meat shares. And this was the largest share on NASDAQ on that day. This is something that you cannot ignore. And we ask ourselves, why are people so buying so much proteins and so interested in proteins and alternative proteins? Is it a matter of choice, sustainability, or health? Well, I even found myself looking at a burger like this one on your screens, trying to consider whether I wanted to taste this Beyond Burger. But even here, this matter of choice you can see of this actually plant-based burger is actually a little bit off, because if you look at the menu, you can see that even one choice is the Beyond Crispy Bacon and Cheese burger that actually entails bacon, which is obviously an animal protein and not vegetarian nor vegan. So we ask ourselves, is it really a matter of choice? Are we really offering a more sustainable alternative? Is it really healthy? Because on that same month when the Beyond Meat share was launched, this was on the CNN front page. Ultra-processed foods are easy, cheap, and could be killing you. So are we offering people good choices, or are they bad choices? Because if you think about it, the ultra-processed food concept that was actually initiated by the NOVA classification of the FAO mentioned that the most important factor today about diets is not nutrients, but rather food processing, defining 4 classes of foods, with ultra-processed foods being named as the most adversely affecting products, with examples ranging of a variety of processed foods, starting from breads, buns, margarines, breakfast cereals, all named ultra-processed foods because of they are highly processed and they contain many ingredients and isolated ingredients. But if we go back to the Beyond Burger and we flip the cover to the ingredients list, we see that even the Beyond Burger, which is now sold globally as a good alternative, a sustainable alternative, is actually filled with a lot of ingredients that would easily classify it as an ultra-processed food, which is part of the health trend to try to avoid. So where does this story intersect? Where proteins intersect with our health. The intersection is the gut, and this is actually the main interest of today's talk, because any protein that we ingest will probably go into our gut, where it will face the digestive functions, the neuronal functions, the immune functions, as well as the microbiome, which is an auxiliary function of the gastrointestinal tract, where this food or this protein will find its digestive fate into our body and into the action and the implications to our health. This actually involves a few steps. If you look into the literature, this intersection actually involves the breakdown of proteins in 2 main compartments of the gastrointestinal tract, the first being the stomach and the second being the intestine, where actually the proteins are broken down by enzymes and by hydrochloric acid through pepsin activity, trypsin activity, and other enzymes like carboxypeptidases and chymotrypsin that are secreted from the pancreas, broken down into 3 and D peptides and amino acids, which are then absorbed through the epithelium into our bloodstream, where they are actually distributed throughout the body, where they can be utilized to build muscle mass and to do bodily functions. But how do we understand how this occurs? How do we study this? Well, first of all, we need to understand that not all amino acids are essential to health, because we can actually form some of them, but some of them are essential to our health because we cannot make them. And Studies actually try to understand what happens to proteins in our diet. These actually apply in vivo methods where parameters like protein efficiency ratio, the net protein ratio, or net protein utilization parameters can be measured, whether it is in humans or various animal models, or you can go for the more biorelevant parameters like true digestibility, the PDCAS, which is the protein digestible corrected amino acid score, and the digestible indispensable amino acid scores that are actually used to classify alimentary proteins, and the complementary in vitro methods that are used to calculate these values that take into account the amino acid profile of the nutritional quality of the protein. This is where the Infogest network is actually quite active, and we are trying, part of us, to understand digestive proteolysis and the underlying mechanisms behind it. We can apply a series of in vitro or in vivo methods with varying levels of complexity and biorelevance, but altogether, even the simplest or the static in vitro digestion model that was recently published even as a Nature protocol last year are well correlated with in vivo data. So they actually give us a very good indication into how proteins are broken down and rendered bioaccessible to humans. If we look into digestible indispensable amino acid scores that have been calculated and determined for various protein sources, we can see that the actual gold standard is actually whey. And when comparing the digestibility of whey to plant proteins, we see that plant proteins fall short. They fall short significantly. For example, if you look at kidney beans, or at, if you'd like, even soy or peanuts, which are very protein rich, they fall very short in terms of their digestibility compared to whey, which is an animal protein. But it's not just about the chemical essence, it's also about the complexity of digestive proteolysis. Even back a decade ago, a very good review highlighted the fact that digestive proteolysis is not as simple and straightforward as textbooks would like to mention. So the fact that most of the proteins come in a very complex food system, they can interact with other food ingredients like lipids, like phospholipids, and even with some carbohydrates, therefore affecting the rate and the extent of digestive proteolysis to varying extent. And this has even been verified in various studies, including studies of MRI, magnetic resonance imaging, done on humans. Even our in vitro systems have highlighted the complexity of the proteolytic process. For example, experiments taking whey protein solutions and subjecting them to in vitro gastric proteolysis show that there is a very strong kinetic competing mechanisms there. On one hand, proteolysis is actually breaking down proteins, making them more and more soluble. But on the other hand, gastric pH is changing over time after ingestion of a meal, and this actually can induce isoelectric precipitation of some proteins and clotting of whey, for example, as you can see in these images. Differences between, for example, processed milk and non-processed milk or non-pasteurized milk can be seen both in vitro and in vivo experiments. And what about the comparison of animal and plant proteins? Well, the bottom line is actually at the top of the slide. Overall, plant proteins are somewhere between 40 to 85% digestible compared to animal proteins, which are much more readily digestible in the human digestive tract. Why is that? Are proteins so different? The answer simply is no. But plants do not come as plant proteins. They usually come in a plant matrix, and these matrices also entail other components. that are anti-nutritional factors. These have been recently reviewed and you can see the details of this review in the bottom of the slide, showing the anti-nutritional factors that have been already identified in various plant protein sources. So even if you take carob, peas, legumes, chickpeas, beans, soy, etc., you can find in a lot of these natural sources, natural plant sources, you can find intrinsic anti-nutritional factors that actually limit or hinder the breakdown of proteins, therefore affecting their nutritional quality to the consumer. And this is where processing can come into play, because processing can actually inactivate or degrade some of these anti-nutritional factors or these anti-nutritional interactions. Processing can actually cause various interference with these, and there is a wealth of studies done recently and over 20 or 30 years ago as well, showing the effects of cooking, microwaving, autoclaving, drying, various industrial scale and home cooking methods that actually transform raw proteins into processed proteins that are more digestible to humans. And you can see some of the values for that they have been determined in actual studies. For example, cooking increasing by almost 10 to almost 13% the true digestibility of chickpeas, and you can see the list goes on, and this is just a summary of the list. Obviously, this is not just due to the inactivation of nutritional factors. This is also due to various changes in structure of proteins. So you can form various aggregates, you can form nanoparticles, nanofibrils, microparticles as a result of processing. And obviously, the fact that these assemblies have different architectures render different accessibility of the proteolytic enzymes to actually cleave these proteins into pieces and bits that can be uptaken by the gastrointestinal tract. And what's for dessert? What about trying to find alternative proteins that are not necessarily plant? What about trying to find other animals to eat? Well, the FAO has registered over 1,900 edible insects. In fact, 2 billion people already eat insects globally. You can see a picture here of one of my guests joining me to try out grasshoppers. And in the Western world, this is actually not so well accepted. Actually, it is frowned upon. Insects are considered in the Western world to be dirty. Therefore, the actual use of insects in the Western world in processed foods is actually very, very limited. It's mainly limited because of consumer acceptance and regulatory affairs, because they haven't been consumed in a lot of countries, for example, in Europe. Therefore, rendering them a novel protein in some cases, which actually necessitates a different regulatory path to get them approved as food materials. Processing, however, may help mask at least the biggest hurdle, which is the appearance of the insect, but at the same time also help us unleash the nutritional potential of the insect. But first of all, why eat insects? Well, in the previous slides, I mentioned that plant proteins may be less digestible. Insects are an animal, so they are animal protein. They contain a lot of animal protein. They are highly edible. They are very efficient in converting feed to food, meaning, for example, if you need 1 kilogram of cow protein, you would need to feed it with 20 kilograms of feed. With crickets, the story would be by an order of magnitude less, meaning that for 1.7 kilograms of feed, you would get 1 kilogram of grasshopper or cricket protein. They're highly nutritious, they're eco-friendly, and they're highly edible. So they're a good alternative that we must explore. And this is part of what we've been doing in our group, trying to see whether we can process, for example, crickets in this case, to try to manipulate or to improve their gastrointestinal proteolysis. In this case, we looked at cooking and baking of cricket flour, or what is called a cricket flour product, which is basically ground crickets that have been pre-treated and obviously clean and standing up to all the health codes. The bottom line of this study was that processing actually improved the solubility of the proteins and their digestibility, and this was published back in 2017 in Food Hydrocolloids. We then moved on to explore other insects, and not just other insects, but also other target populations, for example, the elderly population, which actually needs to get a lot of protein in their diet. So we went on to explore the possibility to actually create an insect-enriched ice cream that is actually protein-rich ice cream. So it actually is a very good, comforting, and very comforting food to offer to elderly. It doesn't challenge their dental anatomy. It doesn't. So if they have mastication problems, etc., ice cream is something that's very, very easily chewable. So you don't really need to chew on it. And we even pursued sensorial tests, and you can see some of the results at the bottom of the slide here, talking about the visual appeal, the smell, across 40 different volunteers, getting scores that were beneath normal ice cream that wasn't insect protein enriched. However, this was not statistically different, meaning that even when we added insects, people liked it less, but not to a significant extent. So to come to a conclusion, a great thinker once said, "I think, therefore I am." And today I'd like to extend this great sentence with the thought of, "I cook, therefore I am human." Because when we think about it, we are the only species on this planet that cooks and eats and lives off cooked food and processed foods. Whether we are doing the best or the optimal way of cooking and processing foods, that is a completely different question. And this needs to be answered through the true understanding of the digestive fate of foods. So to wrap up, proteins provide us with more than just amino acids. It's important to understand that we have to balance diet with physical activity and life. So even if we try to reduce, for example, our caloric intake, we will probably not get good weight loss, because we need to have physical activity intertwined into our lifestyle. But the main take-home message from today that I would like you to take with you is that processing helped and will help us unleash the protein potential in plant proteins and also in insect proteins. Because if you think about it, many of the new plant-based foods and all of these novel foods are actually and will be classified as ultra-processed foods. But that does not mean that they should be disqualified. Contrary, this means that they are something new to put on the shelves to actually try to provide people with the right foods, because we have to remind ourselves, especially today with COVID around, that all people have the right for the right food. And we, as food professionals, have to understand this and have to pursue research in the field to try to help us as humans unleash the potential of our environment to actually nourish us to the best possible way. Obviously, I cannot finish without thanking the actual people that help and drive the research. So first of all, the funding agencies that actually provide us the tools to do this, and the actual people who do it, that you can see their happy faces in front of you. And obviously to all the past and present collaborators that have contributed to these studies whether it is actively or passively through very intriguing and stimulating discussions. And obviously, I cannot finish without thanking all of you for your time. Thank you very much.
Thank you, Uri. That's very nice, very interesting. We do have some questions. Myriam, would you mind starting them?
Yes, so the first question is, what happens to bioactive peptides or hydrolysates in the gut and the intestine? Are they broken down further to their individual amino acids, and do they therefore lose their functionality?
Well, in many respects, protein are degraded. They are uptaken, and in healthy adults, the uptake of D and tripeptides is the largest sequence that has been documented to be uptaken. So most peptides are actually screened by proteolysis, broken down and only uptaken through various controls in the gut and the gut lining that actually decides what to take up and what not. One of the other questions that was raised here was about processing and the role of processing, and the fact that it is, on one hand, the necessary evil, if you'd like, because many studies and there's a big hype around ultra-processed foods and the correlation with adverse effects to health. But on the other hand, one cannot ignore the fact that processing helps unleash the potential of proteins, because in their natural form or as raw ingredients, they are in many cases very poorly digestible. And by transforming them into a more digestible form through processing, be it cooking or whatever, we've been able to unleash the calories and the energy and the ingredients that are in there, whether it is by inactivation of anti-nutritional ingredients, like lectins and polyphenols and other ingredients, tannins, that can actually interfere with proteolysis. So processing is something that we cannot go and think about it linearly. It's not like going 0 or 1. We need to find an optimal point for processing, and this is part of our responsibility as researchers to try to help the scientific community give answers as to what is the right way or the best way to process foods for optimized health. Another question was about insects and the target populations that we've been discussing in this seminar. Well, insects are not that bad tasting. In fact, even some sensorial tests show that you can mask off their appearance and their unique attributes by mixing them with foods, like many food manufacturers already do, for example, with iron, or for example, in many yogurts with flavors. So it's definitely something that is possible. And more than that, it is something elemental in incorporating insects into the Western diet by diminishing the insect appearance, which is the biggest hurdle. Another question that is coming here is about the concept of ultra-processed foods, and that is, wow, that is an immense field of debate. What are ultra-processed foods and what are the NOVA classifications, whether they are correct or incorrect, or how accurate they are. Sadly, time will not permit us to go through a different seminar just on this topic. So I invite you all to visit the FAO and look at the definitions and the scientific literature that has followed since its publication almost a decade ago.
Uri, we have about 18 questions left, but we don't have enough time, unfortunately. I would suggest we move on. For all the others, we will publish this webinar on YouTube, and you can ask questions there, and no doubt the presenters are happy to answer. Other people are can respond to any questions. So we might move on to the second speaker. Thank you again, Uri. We might move on to the second speaker. It's Myriam Grundy. Again, it's a pre-recorded talk, and we can you can type in the questions in the question and answer box. After the question and answers, I have 3 more slides. I've 1 or 2 announcements regarding the Food Digestion Conference, so bear with us. So next is Myriam Grundy.
It is my pleasure to talk to you this afternoon on the subject of the structure of plant-based food and the effect on nutrient bioaccessibility and digestibility. So it seems to be like, if you look at social media in particular, and even looking at the literature, it seems to be different opinion about the effect of certain nutrients on health. And this is particularly true for lipids and cardiovascular disease. The composition is often taken into consideration, but not so much the food structure. However, the form of delivery of the nutrients and different bioactive compounds contained in the food is essential, because it will affect the bioavailability and the efficacy of those compounds. And in a food that is more structured will take longer to digest. And this will have some consequences on satiety and potentially on food intake. Also, the site of delivery of those nutrients or bioactive within the gastrointestinal tract is important and is likely to differ depending on the original structure of the food ingested. And another hot topic at the moment is the microbiota. And currently we don't know what is the effect of the structure on the microbiota. So the food structure influences nutrient release and digestibility, and subsequently it affects nutrients absorption and the energy metabolized. So if we look at the organization of 2 specific plant-based food, so starting with almond, so we ingest the almond either as a nut like that, or under the form of flour or marzipan. So the particle size in this case is reduced. And mastication will also play the same role and reduce the original particle size of the almond. And this will lead to particles that will contain a range of cells that are ruptured, and sometimes cells are separated. And those 2 scenarios depend on the structure and the property of the cell wall. For the almond, we know that a cell will rupture under pressure. And so you can imagine here, once the cell wall is ruptured, the nutrients will get released and then will get digested. Anything that escape the upper part of the gut will reach a colon where the material will get fermented by the microbiota. So another scenario is the oats. So similarly to almonds, you need to process the oats in order to be able to consume it. The most common form of consumption of oats, at least in the UK, are oat flakes. And you can see that depending on the form of processing, the structure of the plant tissue and the level of degradation of the cell wall and the dye fiber will differ. And this will have, again, consequences for the digestibility of the nutrients contained within those foods. So the main question I'm working on at the moment is trying to understand the effect of that structure of the food and processing as well, and see the consequences on nutrient release and digestibility. And what we can see already is that different plants behave differently, and there are different mechanisms that can explain the positive impact that certain food may have on health. So nutrient bioaccessibility and digestibility have implication on the risk factor of cardiometabolic disease. These have been well studied and documented. Plant food structure, and in particular, so the data fiber, the cell wall, has an impact on nutrient release and digestibility. And the mechanism explaining this are several. So an encapsulation of nutrients is one of them. Increase in viscosity in the intestinal but also gastric content. The interaction of those data fiber with different agents of digestion by salt and different enzymes. And finally, interaction with the mucus layer. Other compounds also present in a plant food matrix can also have an impact on nutrient digestibility and metabolism. So polyphenol are an example, phytosterol and anti-nutrients, in particular for protein. Just a quick reminder of what data fiber are. So starch, under certain circumstances, can be considered as data fiber. So this is true when they resist digestion in the upper gut, and because, for instance, of encapsulation or lack of gelatinization, those starch will reach the colon and will then be degraded there and behave as data fiber. But the main type of data fiber based on the codex definition are the cell wall components. So they are made of different polysaccharides. So the three main one are cellulose, hemicellulose, and pectin. So now looking in more detail at some of the mechanisms I mentioned earlier, starting with encapsulation of nutrients. So certain plant-based foods have a cell wall that need to be broken in order for their content to become available for digestion. So the cell wall are in general not permeable to digestive enzyme unless they have been ruptured and damaged. So this is true for almonds, for instance. And if you look at picture of almond here, so in the center of the particle, you will see that the cell wall is intact, and the majority, so all the nutrients are still enclosed within the cell. On the other hand, at the edge of the particle, you see that the cell has been broken and the content has been liberated, and potentially those nutrients can be then digested. So looking under the confocal, when we separate the cell, we notice that intact cell wall will contain here, shown in red, some lipids are still intact and undigested. On the other hand, when the cell wall seems to have been damaged, and we label here in green some lipase, we can see that the lipase can penetrate, and the coalescence of the lipid shown here demonstrates some digestion taking place, but still the digested product still enclosed within the cell wall. We then look at the digestibility of almonds of different particle size down to separated cell, and we use almond oil emulsion as a control. And we use a pH stat to follow the digestibility of the lipid. And what we notice is, like, for our control, so the emulsion, we see a rapid and total digestibility of the lipid. However, when we increase the size of the particle, we see a decrease in the rate and extent of lipid digestibility, down to the cell, separated cell, where the extent of lipid digestion is pretty low. So this demonstrates the encapsulation phenomenon occurring in almond. Now, looking at polysaccharides that are soluble in water. So those polysaccharides will leach out in presence of water or in presence of fluid from the gastrointestinal tract. And this solubilized polysaccharide will affect the viscosity of the GIT content. And this will have some consequences for nutrients digestibility, as it will affect the mixing and the chance of the enzyme to come in contact with the substrate. So cell wall porosity can also be altered as a consequence and become permeable to digestive enzyme. And processing will amplify this behavior, particular cooking, as we're going to see in a few moments. So oats is this kind of plant-based food that contain a lot of soluble cell wall polysaccharide. So looking at the digestibility, so here in this diagram, you have digestibility of starch, lipid, and protein. So this is the original material, so the content, 100%. So we then looked at flakes and flour, is raw and cooked. And we noticed that for the raw flakes, the digestibility of all nutrients is rather limited. And once we cook those flakes, the digestibility is greatly improved. To improve even further the digestibility is making a flour. In this case, raw, we're having a similar digestibility to the cooked flakes, but cooked flour, it's even better, with 26% of the nutrient being digested. And if we look at what we recover after digestion, so for the raw material, we notice that a lot of cell wall are still present and seem intact. And the starch, because it's raw, it hasn't been gelatinized, and therefore they are not digested, which is translated here with this high recovery of starch after digestion. On the other hand, when we cook the oats, we notice that more cell wall get degraded, and some matrix are formed. So we're not quite sure exactly what it is, but we suspect it will be some kind of network formed between the protein and maybe some remaining of the starch that hasn't been digested, and maybe it's part of the cell wall as well. So what about other plants? So I talk about almond and oats, but we study as well amaranth. So amaranth is a staple food that is found in particular in Kenya. It's quite underutilized in Western country. However, it's rich in protein, dairy fiber, and macronutrients, so it could be a really interesting source of nutrients. So this is how an amaranth plant looks like, and the seed looks like that. So it's really tiny seeds that look a bit like mustard seeds, so in terms of size and shape. And what we were interested in is looking at the effect of processing on the nutritional value of those grains, so in terms of composition, but as well digestibility of the nutrients. So we use amaranth under different forms. So we looked at the original grain. We looked as well at popped amaranth. So this is a bit similar process than popcorn. And this is highly consumed in Africa and as well in South America. And we looked as well as at flour, and we isolated the oil bodies as a control. And what we noticed is, it wasn't really surprising, but either the raw or the toasted grain show really low digestibility. And as soon as you grind those grains, you see an improvement in lipid digestibility, in particular for the raw flour. And looking at the oil bodies, we saw again high rate and extent of digestibility. However, we were a bit surprised to see that puff amaranth wasn't more digestible than that. But actually, when we think about the process, the cell wall is still mainly intact in those grains, and a lot of nutrients are probably still enclosed within the cell wall and within the cell content. So looking at the microstructure of the amaranth grain, so you can see on those confocal images that the amaranth grain is really small. Like you can see the whole grain under the confocal. So here we're having the perisperms that contain most of the nutrients. So protein here stained in red, but you will find also the starch. And you can see here the cell wall in blue. And this part here is the endosperm that contain some of the lipids. So you can find the endosperm here again in that images. And in green, you clearly see the lipid. You can also see a layer of lipid over here, just underneath the hull. And when we look a bit more closely at that endosperm, we notice that the cells have a different shape and are much smaller than the cell from the perisperm. So this may explain why, unless the cell wall is broken, the lipid will not be easily accessed and digested by the enzyme. So just to give you a brief overview of cell wall characteristic of different plants. So I talk about almonds. So here you can see separated almond cell. Oats. So here you will have a picture of oats tissue from Wood and Fulcher from 1978. And I can't show you any separated cell for oats because it's really difficult to separate. It's almost impossible to separate those cells due to the property of the cell wall. Here you having amaranth, and here you having lentils. So you can notice, first of all, that the size of those different cells varies, with lentil in particular and pulses in general have cells around that size between, let's say, 100 and 200 micrometers. So they are much more, they're much bigger. So you can imagine during processing they will behave differently as well. I give you some estimation of the cell wall thickness as well. And so those three plants will have cell with cell wall with a thickness quite similar. On the other hand, for the oats, we've got great variability based on the cell you're looking at. So the cell wall are much thicker in the aileron and sub-aileron layer over there. And the data fiber content is slightly different depending on the variety you're looking at, but they are within the similar range. But what is interesting is to look at the type of data fiber. So we can classify them based on the solubility in water. So almonds, amaranth, and lentils are pretty rich in insoluble fiber. On the other hand, oats contain more of the soluble type, and in particular the hemicellulose, beta-glucan, and to a lesser extent arabinoxalant. So you can imagine that because of those structural features of the cell wall, and because of the composition as well, their behavior during processing and digestion will differ. So to summarize, cell wall play a critical role in determining the behavior of plant-based food during processing and digestion. When the degree of complexity of the material increases, the nutrient digestibility is reduced. A significant proportion, however, of nutrients remain encapsulated after in vitro, and haven't shown any data here, but we show the same in vivo. So after gastrointestinal digestion. However, the process that impact nutrient digestion differ based on the plant that is studied. So I like to acknowledge all the people who work with me to get those data. And I like to thank you as well for your attention.
Thank you, Myriam. That was an excellent talk. Very nice. We lost Myriam along the way at some stage, so I'm glad that we pre-recorded the talk. So I'm actually relieved about that. Myriam, you have one question?
Yeah, thanks, Myriam, for a great talk. So the first question that came in was, apart from resistant fibers, what happens to the food structure in the gastrointestinal tract? Does the very low pH in our stomach make all food structures irrelevant?
Actually, this is quite an interesting question, but it really depends on the food you're looking at again. So in the example, when I show different dairy fiber and resistant starch in particular in potatoes, there will be still a release of those resistant starch at some point in the GI tract as the digestion takes place, because the cell wall of those potatoes is more, can leak, possibly can leak. Again, I haven't studied potato in details, but I suspect the cell wall can degrade much easily than some other plants. So the other example I know much better is the almonds. In this case, the cell wall will be resistant, and even the low pH in the gastric environment will not have much impact basically on the cell wall component, and will resist digestion through the gastric environment, the small intestine, down to the colonic environment, where fermentation will take place eventually.
Excellent. Again, we have 9 questions, so I just pick out 2 more. One question is from Philip van Buxtelen. Digestibility of oats increases by cooking. Is it a cell wall effect or just mainly the cooked starch which is accessible for digestion?
So a bit of both, actually, because obviously, as I show on those microscopy, and is well known, if you don't gelatinize starch, you will not digest it. But both by cooking, you will increase as well. The cell wall will swellen and absorb some water, and some of those polysaccharides will leach, while the starch will as well gelatinize, and so the accessibility of the amylase will be increased. So different mechanisms taking place there.
Very good. And I have one short question. Do you know if non-thermal techniques have been used to enhance digestibility of plant nutrients? So non-thermal techniques.
Repeat.
Do you know if non-thermal techniques have been used to enhance digestibility of plant nutrients?
I suspect, according to what I read, probably extrusion will be a technique by actually degrading completely the cell wall, breaking it by mechanical pressure. Fermentability, maybe using some yeast or microorganism instead of cooking to increase, so to degrade some of that food structure and some of the cell wall as well.
If I can add, there is, this is Uri. There are 2 or 3 papers out in the last year or 2 talking about high pressure processing, pulsed electric fields, ohmic heating, and other, these emerging technologies, the effects they have on various plant proteins. So you just need to look those papers up. I think I saw some of them online.
Excellent. So we might move on. We have 3 minutes left. I have three more slides. So there is an announcement. The local organizing committee for the next Food Digestion Conference in Cork, and that's led by Linda Giblin. And the steering committee, the Infogest steering committee, has decided to postpone the next Food Digestion Conference to May 2022 for obvious reasons, unfortunately. So we hope you will make it in 1.5 years' time. So in the meantime, we have to live with our online world. That's why we organize these webinars. And because of the first webinar, more than 500 people signed up, and really from around the globe, from Europe, North and South America, from Asia, even some from Australia and New Zealand have signed up. At the moment, there are over 300 people have joined. We will put this up on YouTube, so watch that space. We will also organize more webinars, and we decided each first Wednesday of each month, 2 o'clock Irish Standard Time. So the next webinar will be 7th of October and then 4th of November. Again, 1 hour, 2 speakers, probably one senior speaker and 1 junior speaker, covering topics of relate to food digestion. And again, follow that space on LinkedIn, on the Infogest group, or Infogest emails. I would also like to share this with you. It's a mini symposium that we are organizing. We had a food structure and functionality conference planned for June, and that is also postponed to October of next year. But we are organizing a 2.5-hour mini symposium. Again, it's an online, free symposium. We have some terrific speakers there. As you can see, Julie McClemens, Milena Kortik, Anvisha Sakar, our own Laura Gomez-Mascaraca, and another speaker on in vitro meat. Again, slide here, how you can follow us either on LinkedIn or Twitter or email. Until then, I hope to see lots of you in person in Cork, and until then, hope to join us for our next webinar, 7th of October, same time. Goodbye, Slán.